students. I'm sorry for the delay for this. I've tried this multiple times, and I'm just gonna go finish this up. And if I needed to add more explanations on Monday, I will. But also, I will post additional course materials for this particular chapter. Anyway, this is an image I've shared with you a while back when we were doing the introduction of the nervous system. These are the levels of organization, and we pretty much covered all of the boxes here, from central nervous system all the way to the last two boxes of sympathetic and parasympathetic nervous system. So today we are going to focus on the sensory or afferent division. And although we have an understanding of the general physiology of that and also structures, what we're going to talk about today are the sense organs. Organ is the eye, which is for the sense of vision. Again, this is one of the first images I shared with you, and it just so happens that the first function of the nervous system shows you an image of a glass of water being focused in the eye and will be transported to the brain for integration, and then respective motor output will of course be followed by that. Let's look at the out of the five senses that we have, 70% of all the sensory receptors are in the eye, which I think is amazing because the eyes are small compared to the size of our other sense organs. I mean, the skin alone makes 8% of our body weight, and yet most of our sensory receptors are found in the eye. So we know of five senses, but there is, most scientists actually consider them to be seven, and I agree with that. It's just that the five are the more popular ones. So I'm going to share with you a website in Life Science. It's a very short article about the seven human senses, and I think some of you are already familiar with the other two, but take the time to read that. It's a very short article and just an overview of the different senses. So, nearly half of the cerebral cortex is involved in vision. So what that means is it's really not just the occipital lobe, which we said was the primary visual cortex. When we were doing the mental status examination, the process of visual information is also has a focus in the other lobes of the cerebrum, the parietal, the temporal, and the frontal lobe. So obviously the eye is this image right here, which is sort of a review of our skeletal system. So we can see here the orange would be your frontal bone, but also your ethmoid bone right in the middle. You have the sphenoid, the lacrimal, the maxillary, and the zygomatic bone, which borders our orbital cavity or the eye socket. Protection. The upper eyelid, the lower eyelid, which protects the anterior of the eye, which is the one that's constantly exposed to our outside environment, which could be filled with irritants. And then we have the hairs of the eyebrow and the hair of the eyelashes. So they are for protection. In this case, in entropion, where the eyelid is turned in, the eyelashes are actually causing irritation to the eyeball and not offering protection at all. On the bottom image, that's ectropion. The eyelid, which is supposedly, again, protecting the eye, is turned outward, and we can see this as a common anatomy change when we age. The eyelids turning out, that also makes our eye more prone to irritation. That is a procedure called blepharoplasty, which is also the same term we use for the repair of the eyelids for cosmetic purposes. So let's a different view of the eye, showing you structures in the sagittal section. If you remember, we've had the muscles of facial expression. So the levator palpebrae superioris lifts the eyelid, the orbicularis oculi muscle allows you to squint or close your eyes tightly. We can see also layers and layers of fat for protection. And then we have the tarsal plate, which provides support and firmness of the eyelid. In the tarsal plate are modified sebaceous glands, also known as meibomian glands, also known as tarsal glands. So, being sebaceous glands, they secrete oil, which of course adds to protection by keeping the moisture in the eye. So, there's water, but in order for that to not evaporate and dehydrate, then we have a film of oil secreted by tarsal glands to keep it moist. And then we have the conjunctiva, which is the inner mucous membrane lining the inside of the eyelids, but also the outside, extending all the way to the outside of the eyeball. Clear view of your conjunctiva. Again, inside of the eyelid to the outside of the eyeball. This one shows you that the conjunctiva is inflamed, conjunctivitis. So tear glands or lacrimal glands are other protective structures of the eye. Where do you think is it located? Is it medial or lateral? Most people will think it's medial because the tears run down towards the medial aspect. But it's actually not. It's in the superolateral aspect of the eyeball, which makes sense because, well, tears need to flush all of the eye from outermost to innermost. Imagine if your tear gland is on the medial side, then it's just going to run down straight to those little canals known as lacrimal canaliculi and into the nasolacrimal duct. So we produce tears, washes of the eye, and then drains into the nasal cavity. So you could just imagine if there's an obstruction in the nasolacrimal duct, whether it's scarring, a congenital defect, or a tumor, then you'll not be draining into the nose, but constantly tearing out. Even if there's no strong emotions, there's no irritation, we constantly produce tears, but we don't see us tearing because it's drained in the nasal cavity. Other pathology, well, not pathology, but a procedure to correct a certain pathology known as dry eyes. So for people who are not able to produce enough tears, we need to keep the most tears as we can in the eye to keep it moist. So we should not allow it to drain constantly. And in this picture, they have an implant in the punctum that leads to the canaliculus that drains tears from the eye. So we block that so we keep whatever tears are left and keep the eye moist. This is a lateral view of the eye showing you the EOMs, also known as the extraocular muscles. So they all insert in different aspects of the eyeball, allowing them to move the eyeball different directions. And we did this when we did cranial nerve testing in class. And as you can see, there are many, many actions that different eye muscles are able to do. And on the right side of that table are the different cranial nerves. Our main one, of course, is cranial nerve 3, but we also have 4 and 6 that move the eye. That would be the superior oblique and the lateral rectus. Going back, here's the full view of the structures of the eyeball. A little eyeball has so many labels for us to do. Let's focus on one right there. The wall of the eye contains 3 layers. The white sclera covers the entire eye, but on the anterior side, it actually becomes the transparent cornea. And it has to be transparent, otherwise we'll all be looking like zombies. So it's transparent so we can see the inside, what else is inside the eye, which is the pigmented iris, the pupil, and so on and so forth. So let's move on to the middle layer, the vascular choroid, more appropriately colored red because, again, that's where the blood vessels are. And the yellow retina is the sensory layer of the eyeball, and that's where you have receptors, which are your rods and your cones, which are which have a concentration in an area of the eye known as the macula lutea, a yellow body, particularly in that concave area known as the fovea centralis. There's also a part of the eye that doesn't have any retina in it, or that doesn't have rods and cones in it, and that is called as the optic disc. And because there are no sensory receptors, it's also known as the blind spot. I would like to talk more about the rods and cones. If we were in class, I was going to show you these three selected videos. The first one would be a short video explaining the difference between rods and cones. The second one would be the fovea centralis. And then the third would be visual field processing, the visual system. So please take the time to view these short videos. They're only about 5 minutes each. And if you have any questions, just write them down and bring them to class on Monday. Here is an image showing you one of the emergencies in ophthalmology. Can you just imagine if your retina detaches from the walls or tears because of, injury, for example, one of the more common causes of retinal detachment. Go back to the structure of the eyeball and the anterior portion. So there are two chambers or two segments of the eyeball. The one in the posterior side, and in this picture, the one on the right, is your posterior chamber or posterior segment, and it's filled with fluid known as vitreous fluid or vitreous humor. On the left side is the anterior chamber or segment, which contains aqueous fluid or aqueous humor. So that's posterior and anterior, and they are separated by the crystal clear biconvex lens. So that lens is just behind the pupil, and that pupil is in the middle of the muscular and pigmented iris. Pigmented, it's the one that gives the color of our eyes. In this case, it's blue. And as you can see, the middle pupil is black. It's really just a black hole. And posterior to that black hole is a crystal clear lens. Is an ophthalmologist using his ophthalmoscope to view through the pupils, and they're able to see the retina. And this is what's an example of a view through an ophthalmoscope of a normal retina and an abnormal retina. This is not a black hole, but you can see in the pupil this is cloudiness. So this is a dilated pupil, and with dilation they're able to see this white milky substance that's just discoloring the lens and therefore blocking the entry of light into the lens and into the retina. This can be caused by a lot of factors. It could be aging, it could be diabetes, or congenital, which is sad. Here's a baby with cataract and is not able to see. Fortunately, we have it's surgery. Step one shows you a surgeon making an incision into the transparent cornea, and then another incision into the capsule of the lens. And once they're inside the lens, they will remove the diseased lens, the cloudiness, and then they will implant. In step 3, they will implant a new crystal clear lens into position. I think it's a really amazing surgery. Here's another pathology called a glaucoma. Although glaucoma literally means a gray tumor, it really is not. It has nothing to do with it. Glaucoma is when there is increased pressure in the intraocular. Okay, I missed that. I moved that pretty quickly, but Let me just to add on that, there could be increased intraocular pressure because again, there's the inside of the eyeball is a space filled with fluid, and if there's too much fluid, then of course there will be too much pressure, and that pressure can damage the optic nerve. So feel free to go back to slide number 28 and see that pressure damage into the optic nerve. So let's look at the circulation of humors or fluid that may lead to a glaucoma. Let's take out here and show you the flow of aqueous humor. So aqueous humor is formed by the structures known as ciliary processes where there are lots of capillaries. Ciliary processes are where the suspensory ligaments are attached from the lens into the ciliary bodies. So humor is produced there and then it circulates, goes up, out into the pupils so that the fluid flows into the anterior segment, and then whatever fluid is in the anterior segment can be reabsorbed into the venous blood by the slita venous sinuses. So there will no there will not be any accumulation or overflow of fluid there. Otherwise, that will lead to glaucoma. However, if there is any obstruction in the sinuses, if there will be narrowing of the angles between the cornea and the iris, then yes, fluid will accumulate, pressure will increase, glaucoma may result. Going back to the eye doctor, we've seen this chart many times before. This is called the Snellen chart. And I wonder what your vision is. Hopefully you could read line 8, DEF, POTC. Your vision will be 20/20. That means you can see that at 20 feet away. And a normal eye can see that at 20 feet away. So if you imagine someone who can only read, say, line 2, F and P, the vision is 20/100, and what that means is that patient can read those letters at 20 feet away, but a normal eye can actually read it even if the individual is 100 feet away. What do you think is the difference between an ophthalmologist or an optometrist? Well, both are specialists of the eye, but optometrists are specialists of measuring the accuracy of vision, checking for problems of refraction. Ophthalmologists are the MDs. Not only are they able to do what optometrists are able to do, but they, of course, they diagnose and treat more serious eye disorders, and they're the ones that are able to do surgery. Speaking of vision disturbances or problems of refraction, we have our first here. You can see a man moving the book closer to his eye so that he's able to see it, because what really is wrong here is that his eyeball is too long, that the focal point is in front of the retina. So he has to bring the book closer to the eye, closer to the retina where it can be focused. This is called nearsightedness, also known as myopia. How can it be corrected? We use concave lens so that the concave lens can move the focal point further back into that very long eyeball. We have here a man moving the book much further away. His eyeball is too short. The focus is going behind the retina, so he needs to move the book further away. This is hyperopia or farsightedness, and it can be corrected by using convex lens to move the focal point forward onto the retina. Aside from myopia and hyperopia, we have astigmatism, and this is caused by unequal curves in the different parts of the cornea and the lens. The cornea, which is the colorless anterior portion of the sclera, has the most refractive index, and then we have another transparent lens which will help in the refraction of light. If the cornea and lens have unequal curves, there are bumps, they're uneven, then light will be refracted into multiple spots in the retina, giving you a blurry vision. This can be corrected with cylindrically crowned lenses, laser procedures, or just implanting a smooth, clear cornea. So that's it. I know it's short. I had to speed it up a little so that the memory of this presentation will not take so much in our computers. I will do some more explaining on Monday, but at the same time, I will be uploading additional resources for you guys, including an assignment that will accompany this lecture. I hope you learned something from that and you enjoy the rest of the week. Thank you.